Okay, so thank you very much for this invitation. I am Marc-Henri. I'm working at Institut Curie, Paris, France, and I will speak about our development in detecting homologous recombination deficiency using optical genome mapping. So I don't need to introduce optical genome mapping here, or OGM. Just that it allows to reconstitute the structure of complex cancer genome, not detecting the nucleotide sequence, but identifying easily all possible structural variants that can be visualized classically on circus plot, where you can see all the subcategory of structural variant, and in the middle, the translocation in purple. So to build this HRD assay, we got access to a training cohort, and this training cohort was a sample from triple negative breast cancer, or TNBC, and high-grade ovarian carcinoma, or HGOC. Those aggressive carcinoma are characterized by the deficiency of the homologous recombination pathway in approximately 50% of the sample, and this feature is very important to stratify those patients for therapy, in particular for treatment by PARP inhibitor. So we use 41 samples, both from breast and ovary, and those samples were previously characterized by our standard method to determine the HRD status, as well as the sequence of the key gene of the HR pathway, such as BRCA1 and BRCA2. So we analyze those 41 samples by OGM, extract all the structural variants, and we subclassify those structural variants according to their size when relevant, and we compare HRD sample with non-HRD sample for all those features. So we had the expected winner, such as deletion and duplication. Those features were already known and described by this landmark paper of Serena and Nixenal, who characterized a large number of breast carcinoma using deep whole genome sequencing and sophisticated analysis. We were more surprised to identify inversion and dup-inv, and these features were increased in non-HRD sample as compared to HRD sample. However, we finally find that this was reported not as inverted duplication, but as fallback inversion 1 year later by Wang and a collaborator in ovarian carcinoma, with exactly the similar results than what we found: higher fallback inversion or inverted duplication in non-HRD sample as compared to HRD sample. The last surprise were to find a similar number of translocation in HRD and non-HRD cases, whereas this feature was found important from the work of Serena and Nixenal, as well as from previous work from the team. Furthermore, just visualizing HRD and non-HRD sample, there were a clear difference if you just look at the center on the purple line that are indicative of translocation, a clear visual difference between HRD and non-HRD. So we defined another new feature that we name isolated translocation, and we define isolated translocation as translocation free from other translocation event for a distance, a certain distance, as compared to a clustered translocation. So we explore all possible distance from 0 to 5 megabase, and we found that around 2 megabase, there were a complete separation of HRD cases as compared to non-HRD cases. And this was, and to visualize this new translocation, we can see all the total number of translocation in HRD and non-HRD case. Those cases had similar number of total translocation, but if we visualize only isolated translocation, you can see much more isolated translocation in the HRD case than in the non-HRD case. And the statistics shows that isolated translocation was one of the main features of HRD, with a complete separation of the HRD sample and the non-HRD sample. We further define a subcategory of translocation that are balanced translocation when OGM retrieve the two reciprocal events of the balanced translocation. And we found that balanced translocation were also massively different between HRD cases and non-HRD cases. In total, we found 5 types of structural variants: deletion, duplication, balance translocation, isolated translocation, and DUP-INV as differential between HRD and non-HRD. And we decide to construct a model for determining HRD status using logistic lasso regression. And the model retains only 3 key features: isolated translocation, balanced translocation, and DUP-INV event. So to test the performance of this model, we got access to a validation cohort, again with breast and ovarian sample with non-HRD status and non-mutational status. So the model predicts the probability of being HRD, and the samples are on the right of the diagram if predicted to be HRD, on the left if predicted to be non-HRD. So what we found is that the performance of the OGM prediction was quite good, with an accuracy of 22%. So, however, with 2 cases with discordant result between the standard method and the OGM prediction. So we further explore those 2 samples. So this case was predicted to be HRD by OGM, predicted to be non-HRD by the standard method, and we characterize this case more deeply, and we found a methylation of the promoter of FRAD51 together with the loss of the wild-type allele. So a standard mechanism of HRD. Indeed, this case was a true HRD case, misclassified by the standard method and correctly classified by the OGM method. For this case that was predicted non-HRD by the OGM and predicted to be HRD by the standard method, we found no mechanism explaining associated with HRD. However, there is no ground truth available, so we don't know who is right. Is it the standard prediction or OGM prediction? leading to an accuracy of the OGM prediction of at least 96%. In conclusion, structural variant signature of HRD was defined using OGM, including HRD and something new, also non-HRD feature. This method was found efficient even with samples with low tumor content. It is a very visual method, and the bioinformatics is easy to implement. However, it needs further validation on larger cohort, and the main limitation is that it requires large quantity of SNAP frozen tumor material. In any case, it definitively helps clarifying the HR status for cases with ambiguous results using other HRD assays. And as last slide, I want to thank Bionano and my institute who support this work, and in the team, Sandra, who did all the bioinformatic development, the OGM platform at Institut Curie, and the hospital who gave us access to the tumor sample and the patient who consent to this study. Thank you very much.